With Tony Hitchcock. He's the technical director at Cobra Biologics, and his presentation, Expanding DNA and Gene Therapy Market. Tony.
Thanks. So what I was going to talk about this afternoon is something that's pretty close to my heart. It's an area I've been working on, I guess, for the last 21 years of gene therapy products. As I started out in this area, I guess, in about 85 as a startup company. I moved from what was ICI Therapeutics to a gene therapy startup called Thorexis. And we started working out on plasmid DNA therapeutics and going into virus areas. And then a company eventually moved into contract manufacturing. And as a company, we're involved in a whole range of virus and plasmid projects. So Cobra, if you don't know it, have 3 production sites. One in in Södertälje, which is just south of Stockholm. One in the UK at Kiel, which is predominantly on virus and plasmid DNA production. And also further north in Sweden at Mapfors, which is a 500-litre fill-finish site. And at the moment we're involved in doing gene therapy products both at the Kiel site and also looking towards going to the larger microbial site. As a company, we've kind of been involved in a lot of novel and interesting projects. So we initially work on bacterial products. So as I mentioned, we actually started out working on plasmids, which is kind of an unusual startup point for a CMO, but also moved into proteins, bacteriophage, and also more recently into whole cell products. And as mammalian streams, we started out actually making viruses rather than proteins, but now cover the whole range of proteins, being de novo proteins, biosimilars, and also obviously MABs. So why should I speak about gene therapy is that it's been a market that's been very up and down, I guess, for the last 20 years. But over the last 3 years, we've seen a huge emergence of successful clinical outputs of gene therapy. So if you look, even this year, there's a huge number of presentations and publications on successful gene therapy products. As you see across the various investment markets, there's a huge number of IPOs going on and significant funding going into this area. And I think more interestingly as well is that you're now seeing a lot of the large pharma companies getting involved in this area and actually bringing gene therapy as part of their portfolios. And I think within the next 2 or 3 years, you'll actually see all the large pharma companies having a gene therapy part of their portfolio. If you look at the actual market, if you look at the recent analysis from Roots, is that it's clearly quite a varied market in terms of looking at therapeutic areas, people going beyond the original gene replacement areas into cancer, infection, neurology, oncology, and as well as CFD. The delivery vehicles is kind of an interesting area, is that whilst you kind of think people might use one specific vector or not, it's quite clear that these are being chosen and selected for their therapeutic uses. So we're seeing people coming to us with AAV projects, still using naked DNA. Lentivirus is clearly an emerging area, but also things like adenovirus, which was the original vectors people started looking at. People are still very much using those, especially in the oncology field. And in terms of clinical phases, we're seeing now one licensed product in Liberia, but we're also seeing a number of projects coming forward for phase 3 and coming out of phase 1. So it's a very interesting area. While most of the products, like everything else, are still in phase 1, we are seeing an emergence of projects successfully coming out of phase 2 and into phase 3 studies. So when you look at that as a market, it's kind of incredible that people are looking at potentially a 20-fold growth in the market over the next 10 years of sales going between maybe $9 to $12 billion in the next 10 years, which I think is an incredible increase. What's also interesting is that it's very much looked that they think 80% of the market's going to be in the oncology area, but also what's not counted within these numbers is potential of combination therapies of people using, especially immunotherapy, in combination for other oncology treatments. It's always interested me why we should be involved as a CMO in gene therapy, is that gene therapy to me is not just a singular product. It's very much construction of delivery vehicles for therapeutic uses. So it effectively allows, in the same way to a degree the large amount the monoclonal area did, allow you to actually develop processes and manufacturing systems, including facilities, that are disconnected from one specific product. So if you're investing in technical development, whether in process, analytics, or actually building manufacturing facilities, it's not going to be dedicated and linked to the success of a single product. So from a CMO perspective, that starts becoming highly attractive, but also from a drug development company, it allows you to have a platform for producing therapeutics for a number of different conditions. So it should, in theory, reduce cost and allow acceleration of those products to the clinic. If you go back in time, is that DNA was very much first started out coming to us as saying it's going to be used just purely as DNA vaccines. That was the original product. I was involved in doing non-viral gene therapy. But now we're actually seeing quite a wide use for plasmid DNA. Viral vectors is something I'll come on to. People are very much using that for transient production systems. We see some companies looking at very novel template systems, both RNA and DNA-based vaccine systems. And also you see some areas of people looking at can you rapidly make proteins through cell-free synthesis. But I'll kind of focus on gene DNA and viral vectors, but it's clearly an expanding market. In terms of the actual naked DNA vectors, which people kind of looked at us as what you might call non-viral gene therapy, is that this is kind of still an interesting area. People work very hard to try and get clinical results out of this, using them both as naked and cationic complexes to deliver. People are now even using this for CF. There's some very interesting results coming out soon, I believe, on using those kind of systems. And from a manufacturing perspective, it's very clear we need to make these products to full GMP. Often we'll make tox materials to full GMP as well as the actual early clinical materials. But as I said, it's an area where people have persisted and managed to get some really interesting data out of that, and now looking to try and scale these systems out from making what might be a few grams of plasmid up to hundreds, if not kilograms of plasmid. I think it has great potential because, again, as I was saying, we're actually making a delivery vehicle, so our manufacturing processes are not going to be linked to any one specific product or customer vector. And that allows us to disconnect development, analytical manufacturing from that product. But also from a regulatory perspective, I think it's something that's emerging is how do you describe plasmid DNA? Is it going to be a well-characterized molecule that gives us flexibility to change manufacturing processes and scale up? Or is it going to be linked very heavily to a specific process? And I think that's an emerging area. But from a manufacturing perspective, it actually creates quite a large amount of challenges of having flexible production scales because we don't know what the clinical uses are. It's clear that you can get big changes of people initially wanting platform systems for making a few grams of product just for early stage clinical trials and then wanting potentially 1,000-fold scale up of that going into larger in-market product. And also that you see people wanting to look at multiple vectors, so you need a flexible system that you can really turn over a large number of different clinical products and flexible platforms and production systems. The processes for plasmid tend to be very much the same. Companies have their own nuances and patents around processes, but fundamentally they're all the same on the basis of their high-density fermentation from E. coli. The stage that's unique to DNA is actually using alkali detergent lysis, chemical lysis, based on the original Birnbaum-Doyle process. People have their own proprietary systems how to achieve that and the speed of that, but fundamentally they're doing the same things. Purification systems tend to be maybe 2 or 3 step chromatography, and again basically using hydrophobic and ion exchange technologies to remove host contaminants including RNA and also different plasmid forms. And formulations, you see a range of that, but it's not hugely different. A lot of people still looking at liquid formulation systems, potentially going into lyophilization and vialing. So the processes, whilst there's some variation between different companies and approaches, fundamentally they're looking at very similar approaches. And I say in scale, it creates us quite interesting challenges of that for the viral vectors at the moment you see people wanting relatively small amounts to do high throughput screening of design of vectors and initial studies into the kind of AAV type lentivectors. The naked DNA, we're seeing at least 100-fold scale in people doing phase 1, 2, and then looking at going through to market manufacturing. The novel area of this is that because in some cases we're actually using plasmids as starting materials for making viral vectors, is to try and understand the global quality systems that people want, whether it's full GMP, are they going to be just counted as traceable processes, high-quality plasmids. It's still very unclear from the regulatory perspective of what they should be called. It was interesting that the EMA recently released guidelines that suggested that plasmid should be regarded as starting materials, and I think that's an interesting concept that the regulators are now beginning to think that out, but how far they've taken that will be interesting to see in the near future. When you come onto the viral vectors, and I was going to kind of stick to the transient systems rather than the self-replicating adenovirus and those types of systems, where we're actually using plasmid as the starting material for the construction of those vectors, it's been very much an emerging market. People have only recently really been pushing this for larger scale, where you're having to make 3 or 4 plasmids before you can actually make your clinical product. So it makes the clinical production programs really quite complex in the basis that you're making maybe 5 or 6 different starting materials for your product. And so it causes some different opportunities from CMOs to try and develop services around that. As I mentioned, the EMA have actually started to describe the plasmids we're using this as a starting material, and what the full implications of that is something I think we've got to work out, is how far that impacts both for early and later stage clinical development. And it's quite clear that different companies and different customers have different expectations for the quality systems within that. And from a company like Cobra, is that I think we're kind of unique in that we have both virus production and plasmid production, so it's something we're very proud of. The viral vectors create some significant challenges for these, especially the adherent systems, is that you have real challenges trying to take large amounts of that. So one thing you see is quite large variabilities in productivities of these systems, but they tend to be in orders of logs rather than just 2 or 3 times or lower. So that creates a significant problem, especially if you're looking at adherent systems, let alone suspension. Clearly, there's issues more of containment of the viruses. Some have come up to BSL-2, so how you actually manufacture them, the facilities disposable of both waste, solid and liquid waste, creates problems. And it's also, from a manufacturing perspective, very much driven us down single-use technologies approaches where there's very little experience of product crossover. How can you segregate products? There's no real accepted limits of crossover for these kind of products. You've got ultra-sensitive assays like qPCR for identifying potential contaminants. So we were a very early adopter of single-use systems for these technologies, and I personally believe you won't actually take those systems out of that. You also have to have processes that really try and work around the actual properties of these very large complexes, in the fact that they're fragile, they're going to be prone to shear damage. They are labile, and you need to process rapidly and sustain that. And the other thing is they're actually very big in some cases, is that when you talk about trying to handle these things, 0.2 filter, you've got something that could be verging on the size of the actual filter pore size. As with the plasmids, the virus production systems tend to be relatively similar in their approaches, in the fact that you have suspension or adherent systems, quite often adherent still in some cases. And the virus production is basically, whether it be a transient production or an infective virus, is going to be an infection stage, stimulate production, followed by recovery stages, and then purification. Purification tends to be now very much going into chromatographic systems, 1 or 2 step purification. And what you have a great advantage here is the sheer physical size of the virus to separate it away from the most of the contaminants. And then looking at formulation and fill finish, and again, formulation of these products is going to be challenging to try and stabilize these products and then characterize the formulated product, especially with areas like aggregation and stability of those products. I think it's something, when I've talked to the clinicians, there's large clinical groups in London who are really looking at these kind of areas, and one thing they've always said to me as a manufacturing group is, you really have to invest and try and develop manufacturing processes. And I think especially the adherent systems where you're instilling cell factories, it's something as a community we really need to look at, develop. Is whilst you see these products being used in the very rare orphan clinical diseases where it's possible to sustain supply of these materials for a very small patient number, if you start looking at trying to scale these systems up, current approaches often limit them to going beyond that, but they can't get into phase 3, can't get into commercial supply. So there's considerable scope for development and improvement of productivities and processing technologies to allow us to go forward on that. I think for a company, we kind of need to look at how you take that forward. We see customers coming to us with multiple vectors, some proprietary, and how we could use them. And it's something as a CMO we have no real control of, is that these are selected on therapeutic approach and delivery strategy rather than on manufacturing technologies and capabilities. But it still gives you the scope for having potential platform processes. You may need to adapt them and change them, but I think it still gives us scope for having a standardized approach to much of these manufacturing systems. It's also in terms of production scale, it creates challenges in trying to understand the disease area and the therapeutic approach and again can make huge changes on dose levels and what scale might be 20 litres, it might be 1,000 litres you're making for clinical production. It's a significant challenge. The other is the analytical approaches that people have, is that whilst people have very good technologies for looking at protein products, the ones for viral and very large complexes is still under development. And I think that's, when you look at the regulatory questions that are being raised, it's very much the analytical areas they're raising rather than the manufacturing ones, as their concern of how you characterize these products, look at stability and functionality of the products. So in conclusion, I think it's a very interesting area. We're actually seeing what this area very much started out as doing DNA vaccines and very limited technologies, very much now going into these kind of disease therapy, immunomodulation systems. From us as a contract manufacturer, it's very clear to us that plasmid DNA is a very critical element of that, not only of the naked DNA vaccine products, but also how we can supply and meet the requirements of the viral vector market. And how we adapt our approaches to multiple therapeutic requirements and approaches. And as a supplier, it's how we can be adaptable and flexible as possible to try and meet not only the requirements of scale, the volumes, but also meeting the regulatory requirements on that. So on that, I'll ask any questions. That's their certainly current perspective. I think what's interesting to try and understand what the EMA actually require as a starter material, and when you look into the guidelines, it's clearly quite variable between early phase and late phase production systems. So in the early phase products, starter materials can be quite loosely defined, but going to later phase products, then they have to be fully GMP, and I think that's going to be interesting how we adapt and change to do that.
What's your opinion of inducible gene switch mechanisms to deliver and help the delivery of your gene component?
It's clearly quite a very interesting approach. It's not something we've had a huge amount of involvement in, but as a contract manufacturer, I think as long as we understand the vector production, then I don't see it's any more different or challenging to the existing processes. And as I was saying, these are effectively delivery vehicles, so what the customer wants to and development company wants to put into them is their selection. We clearly just need to understand the process and any limitations around that and try and deliver to that quality systems.